In this video on genetics, we're going to be looking at a different type of inheritance pattern, and that is with codominance. These are the key words associated with today's lesson, and these are the learning objectives. So again, really similar, we're just picking out the key points that we need to look at. Okay, so let's start by looking at what codominance actually is. It's a form of genetic inheritance where two or more alleles are equally dominant to each other. So we don't have any recessive genes here, they're equal dominance. So both alleles are present in the genotype, therefore both will be equally expressed in the phenotype. And a good example of this are the alleles in the M and N antigens of blood grouping. Now you can see that in this diagram of the blood groups. One person has the M antigens only, one person has the N antigens, and then there's another person that has both M and N antigens present on their blood cells. So when we write down these alleles, we use a similar notation to what we looked at with sex-linked traits, but this time we have the M and N as a superscript. Now, what you should notice here in the case of heterozygotes, that both capital letters are used. We don't use any lowercase letters because there is no recessive traits. They're both codominant. So if we look at the conventions for writing genotypes as a reminder, we know that when we're looking at basic monohybrid crosses, we have dominant alleles represented by uppercase letters, while recessive alleles are represented by lowercase letters. And we do know that it's a good idea not to use letters where upper and lowercase are not easily distinguished. So for example, using big S and small s, depending on your handwriting, that can be a really bad idea. Similarly with W and C. You really want to make sure that the letters look different in the upper and lowercase so that when your marker is looking at your work, you'll receive full credit for your answers. When we're looking at codominance, both letters but for alleles are represented by uppercase. Now, we may not use the same letters, so for example in the case of the MN antigens for blood grouping, it was M and N, and the same letters can be used for different genes if we're looking at the general blood grouping for ABO blood grouping types. So for example, we use the letter I because that's representing blood groups, but because blood grouping is codominant, we have capital A and capital B. We'll talk more about that later on in this lesson. And finally, just as a recap on sex-linked inheritance for X linkage, again we use X and Y's, and we use superscript for the alleles on the X. Right, so the first example we're going to be looking at, which is found in your booklet, is codominance in sickle cell anemia. Now, sickle cell anemia is a type of condition where the red blood cells or erythrocytes, which are normally this donut biconcave shape, end up being sickle in appearance. So you end up with this kind of half moon shape. And you can see that in this electron micrograph, you have got the normal red blood cells here, look kind of depressed in the middle, and then you have these abnormal sickle-shaped red blood cells. Now, with these red blood cells, it really reduces their oxygen carrying capacity and affects the hemoglobin. So, with normal shaped red blood cells, it's the genes that code for hemoglobin that are affected. So, when we shorten hemoglobin, it's just capital H, small b, and because they have the normal phenotype, they get a capital A. But if they have the abnormal phenotype or genotype for hemoglobin, it's HB and then superscript capital S. So when we look at the genotypes for a normal individual, it'll be HB capital A, HB capital A, because remember, this codes for one gene and this is another. If you have sickle cell trait, which means that you are codominant and you're heterozygous, then it will be HBA to represent the normal allele, and then HBS to represent the diseased allele. If an individual has full-blown sickle cell anemia, they will be homozygous, and both their alleles will be HBS. Okay, so your first task is to complete page 18 of your class booklet, and before you move on, please make sure that you check your answers, as the notation when writing out genetic crosses is essential, and you will be marked on that. Alright, so moving on now, we're going to be looking at multiple alleles. So as the name suggests, there can be more than 2 alleles that code for a particular characteristic. And as we alluded to earlier, the ABO blood grouping system is a common example. So we know that there are 3 basic blood groups, A, B, and O. Now, the alleles for A and B are co-dominant to each other, and they will produce variants. However, allele for O is recessive and does not modify the basic antigenic structure of the blood cells, whereas A and B do. So when we represent blood groups for alleles, we use the same capital letter, which is I. However, when we want to discuss the antigenic forms, which are A and B, so these are the antigens present on the surface of the blood cells, we give them a superscript capital letter, because remember, they are co-dominant. So for blood group A, it will be I superscript capital A. For blood group B, it will be I superscript capital B. However, when we need to write the allele for blood group O, it gets given a lowercase I, because blood group O is recessive to all others. So if we had to write genotypes, this is what they would look like. So for blood group A, you can be homozygous and have the genotype for blood group A as I capital A, I capital A. Or if you're heterozygous and one parent has blood group O, or you inherit the O allele, then it will be capital I, A, and lowercase I. Similarly, for blood group B, you have the same convention, and for blood group AB, it's simply I capital A, I capital B, whereas for blood group O, it's 2 lowercase i's and there's no superscript. Okay, so just to make sure that you've understood all of that, please pause the video here and see if you can complete the table found on page 19 of your booklet. Okay, hopefully you were able to do that without any issue. Most of this is, of course, revision from semester 1. So with blood group A, you have antigen A on the surface of the red blood cells and antibody B. So the possible genotypes that you may have are homozygous or heterozygous. For blood group B, it's the reverse. And for blood group AB, you will have both antigens and therefore neither antibodies, and the genotype can only be heterozygous. And finally, for blood group O, you will have neither antigen present on the surface of the red blood cells. However, you have both antibodies, and the genotype is small I, small I. Hopefully you were able to complete that table without any hassle. Okay, so the next, which is found on your booklet, page 19, I'd like you to have a go at completing this Punnett square. Hopefully you were able to work out that in this first box you will have a homozygous genotype, and that gives blood group A. The next one along, we will have blood group AB, and this will be heterozygous blood group A. Now if we continue that along, you can see here that the majority of the blood groups are going to be A or B or AB, and there's only a single chance of inheriting two recessive alleles and producing blood group O. So the Punnett square is used again. The conventions are the same as what we've seen previously. Now, if we have a look at an example using blood groups, we are told a mother has blood group O, and she has two children that are blood group O and B. So we need to determine the genotype and phenotype of the father. So the first thing we are told is that the mother is blood group O, so that's two lowercase i's. We don't know the genotype of the father, and that's what we want to work out. But the clues that we are given are that the children are blood group O and blood group B. So we know the phenotypes, which I've written in these boxes. So we understand that if the mother is blood group O, she must have given at least one recessive allele to each of her children. We know that if one of her children is also blood group O, he must have or she must have been given the recessive allele from the father. And if the other child is blood group B, that child must be heterozygous because he has received or she has received that recessive gene from the mother. Therefore, we know that the father must be able to produce children with blood types O and B, and therefore he must be heterozygous and have both alleles. And we have proven that by completing our Punnett square and working backwards. Right, so your second task to finish this lesson is to complete the problems on pages 18 to 23 of your booklet. Please make sure that you work through each problem using the correct notation and taking the time to show all your working, as this is where you will be picking up the majority of your marks. Please check your answers to make sure that you have achieved the correct answers by showing all your working. And once you've done that, please complete the chapter review questions to consolidate your knowledge.